Hi all, welcome back to Microbiology lectures, and today we're going to start talking a little bit about viruses, viroids, and pyrons. So from a microbiology perspective, even though biology means study of life, we do study things that are agents of disease, like viruses, viroids, and pyrons that are not alive. And so, actually, in microbiology, these are very important to the design of medical treatments, such as antivirals or vaccinations, and then treatments of like how do we sterilize things, etc. Because a lot of these organisms are pathogenic and disease-causing organisms. So. Let's dive into it a little bit. It's just going to be a kind of a brief overview of the viruses and viroids and pyrons. So first off, let's talk a little bit about the first virus that was discovered. That's tobacco mosaic disease. Okay, it's called mosaic disease because on tobacco plants and other plants, so it can affect tomatoes and peppers and other leaves of other plants that we cultivate. You'll get these spots of different colors, sometimes brown, yellow, different colored spots, and that's the virus that's attacking the flesh of whatever plant it's landing on. It was discovered in the 1890s in a couple ways. So at first we were interested in looking at the organism that might be transferring this, and so at first many times it was thought, well, this could be a bacteria that was transferred by maybe insects. But the nice thing about tobacco is because this occurs on tobacco, and tobacco produces its own insecticide, which is called nicotine, not very many invertebrates will consume the material. And so because we started to see these diseases that were on tobacco, a lot of biologists started thinking, okay, well, maybe it's something microscopic. A microorganism must be the causative agent. And so two influential scientists kind of dove into this. Owansky, who was the first individual to filter out viruses, and did so using a filter, a porcelain filter, that would filter bacteria, the smallest known bacteria of about 0.3 nanometers, or sorry, 0.3 micrometers, and would filter those out. And again, he would take the solution from that filter, apply it to tobacco plants, and they'd still get disease. So he knew that it must be smaller than the smallest bacteria, and Bacteriophages came along and actually labeled it virus, and also did similar work in filtering out viral agents. And so together they kind of coined the term, or started the field of examining viruses. And then Twerton and Hiralel discovered filterable viruses that could destroy bacteria, otherwise known as bacteriophages. And so they were the first to find that when you apply a solution with this filterable virus, that it would destroy all the bacteria, and they happen to be bacteriophages. And then the term virus was coined again by Bacteriophages, which means poison, and we've held the term virus ever since. When initially examined, Twort, Hiralel, Owansky, and Bacteriophages, they all realized that the organism was non-living, or at least they felt the organism was non-living. It had some living properties, but in general it was not a living organism. And due to this, it was originally classified, and still pretty much is classified, as a agent, either pathogenic agent, or a complex series of complex chemicals. And so we're going to talk about what viruses consist of, made of, and kind of how classification is loose, I guess. Classification is not nearly as solid as you would if you're classifying a living organism, which we use Linnaean classification to classify living organisms. We also use a version of that to classify viruses, but still the classification is pretty loose, and many rules are not followed. Later on, after electron microscopes were invented, we could take pictures of the viral agents that cause tobacco mosaic disease, and this happens to be a picture of that. And these are helical viruses, or they form a helix, long rod-shaped viruses. So the protein itself, the coating of the protein, is a helical protein, and then the genetic material is within that protein coating, or that capsid. Okay, so let's look at some other things. Viruses in general, they all contain genetic information, DNA or RNA. To my knowledge, there's never been one found with both DNA and RNA. But that being said, we have a lot of work to do on finding viruses. We found far more bacteria than we've ever found viruses, and most scientists assume that many of the bacteria will have their own viruses that kind of evolve with that bacteria. And so we're probably far underestimating the number of viruses that are out there. They are considered inert particles, or inert entities, because they have no metabolism, so they're not alive. They have no metabolism. They cannot replicate on their own, and they have no motility. So they don't move, they don't replicate, and they don't utilize energy without the help of another organism. Right? When they have the help of another organism, they are genome hijackers, and so they hijack the genome. They utilize their DNA or RNA to hijack the genome of another organism, bacteria, archaea, fungi. I mean, viruses can affect many different organisms, eukaryote and prokaryote, and they'll take over the genetic material and start replicating their genetic material. In addition to taking over the genetic replication, they also take over the metabolic processes in many cases. So they generate energy that's needed to build new proteins, put those proteins together. So the energy that's needed for replication is utilized. And so inside the cell, they really hijack a lot of the processes. Outside the cell, they're inert. They do nothing. And so this is what one of the main components that makes them not alive is they have no function outside of the cell. When they get into a cell, then the replication and the activities are utilized, and it looks like the virus is an organism, because they can self-assemble their proteins, and lots of things are occurring inside the cell. So they're not organisms, they're infectious agents when it comes down to it. Although this is argued many times in many classrooms, and many professors argue the classification of viruses. So just keep that in mind. Some people do think they're alive. Others stick to the idea that they are not organisms, they're just infectious agents. So they require live organisms, can't be grown in pure culture, that means. So when we were doing pure cultures, say in lab, and you played out a pure culture of E. coli, that is a single species on a plate. Okay, pure culture. You can't do that with viruses. You have to have some host cell with the viral agent. And so whether that be a eukaryotic cell, whether that be a prokaryotic cell, you never have an actual pure culture. But they grow great in laboratory situations, and they have been utilized in labs all over the world for many different processes, looking at infectious agents, but also for the purpose of DNA replication. We originally replicated DNA, strands of DNA that we wanted to replicate many, many times, like the process of polymerase chain reaction, which we do now. We did the same kind of process by hijacking viruses that affected bacteria, and because bacteria can multiply, every 20 minutes, you can get a doubling of population. If you take a virus and have that virus has a sequence of DNA that you want copied, then that virus will affect multiple bacteria. Those bacteria will copy that genetic material, divide, and essentially copy your DNA that you're interested in, divide, and keep doing this over and over and over again. So over 24 hours, 48 hours, you've made enough copies of your genetic material, and that's really how we started examining DNA, because DNA itself is, a lot of the DNA that we're interested in is just small segments of DNA. I can't see them with a light microscope, but sometimes you can see the effects of them with a light microscope. So you can see bacteria that have been lysed, that are destroyed by viral infections. But again, with electron microscopes, we can get an idea of what some of these viruses look like. But some of them are still really small. In an electron microscope, it's hard to know all the features of it, even with an electron microscope. We have kind of a couple different ways at which we classify viruses. So we used to classify them on whether they're affect prokaryotes or eukaryotes, and where they be found. Are they found in the lungs? Are they found in the intestines? Are they found in the blood? What is the causative agent? Where's the agent found? What's it hijacking? What cell types is it hijacking, etc. Now, though, with the discovery of more and more viruses, we're starting to group them as if we were going to group living organisms on a Linnaean kind of way of naming things. And I'll show you this in a second, okay? But nonetheless, they can enter cells in a couple different ways. So the agent that's going to enter, okay, the capsid with genetic material that enters the cell is called a viron. Okay? And it can enter the cell by receptor-mediated entrance. So, in other words, the spikes that you see here on the viron would match the receptors of the cell that it's going to enter, and in doing so, it would be engulfed, it would be phagocytized, brought in, and then the genetic material can be transferred into, say, a nucleus, if it's a eukaryotic cell, and that can get incorporated into the DNA of the organism, can be replicated, and translation of that organism's proteins, that virus's proteins, would happen, and then those units can be put together outside of the cell, they can be put together inside of the cell. There's lots of ways in which this can occur. Most of the time it's done inside of the cell, and then they lyse out, or they move through a pore structure out of the membrane. Okay? The other way is for the viron or virus to not enter, but rather inject. And so they are going to inject the viral DNA or RNA through the membrane into whatever the organism, whatever cell it is, okay? Typically bacteria. And then that viral DNA would be incorporated in the genome or not. And so if the bacteria itself can reject it, so this is kind of how we found CRISPR out, the bacteria can reject it or cut it up or dispose of it, remove it, then it will reject the viral infection. If it doesn't, then it gets incorporated, and then two kind of processes can happen. Either the viral DNA can be made and the virus is made, or it can lay dormant for a while. We'll come back and talk about those situations. Okay, when we talk about viruses in general, we call them obligate intracellular parasites. They're parasites because they are hijacking the genetic material, or the ability to recombine or go through copying of genetic material, and they use all the machinery of the cell, including things in eukaryotic cells like the Golgi apparatus, like the endoplasmic reticulum, to generate the proteins that are needed to rebuild the virus. And so viruses that infect bacteria are often called bacteriophages, or sometimes just phages. And again, these were really important for genetic research. Now we use them all the time to look at zoonotic diseases, transfer of genetic material from one species to another, and they can be extremely important from a bacteria perspective, because it also allows for bacteria to have horizontal gene transfer. And so also, as of kind of lately with lots of antibiotic resistance bacteria strains, and the fact that we're not developing a huge amount of new antibiotics, there is kind of a new process of thinking, potentially using these bacteriophages to attack antibiotic resistant bacteria. So if you know of a phage that can destroy a antibiotic resistant bacteria, then we could potentially inject an individual with that phage. That phage would take and destroy that bacteria, and then when there's no more of that bacteria, that phage can't replicate anymore, and eventually over time your immune system would most likely destroy the protein coating of the phages and remove them from the system. You recycle them and use them in building other things. Concerning, obviously, if the virus can adapt and potentially move to a different host of bacteria. But nonetheless, that is kind of a medical process that we're kind of exploring. So just to put it in perspective, okay, human red blood cells are about 10,000 nanometers in diameter. The smallest virus is poliovirus. Well, one of the smallest viruses is poliovirus at 30 nanometers, and the biggest is mimivirus at 800 nanometers. So viruses are really, really small, hence the inability to see them with a light microscope. Many of you have looked at bacteria under a light microscope, and even fairly large bacteria under a light microscope are still kind of hard to see and see much for structure wise. Okay? So that's just to kind of put them in perspective from a size, from like the relative size to other things that they might affect. Okay, so next time we come back, we'll talk about the structure and kind of how we classify viruses.